My name is Minhajul Islam from group number 324. My topic is about antibodies and antigens from the book M.V. Wolkenstein. First of all, the protection of an organism from foreign biopolymers and thereby from infectious organisms, it is accomplished through cellular and humoral immunity. In the second case, immunity, it is determined by the interaction of antibodies, specific proteins produced by lymph cells with foreign biopolymers, which are called antigen. In this case, the immune response, that is the appearance of antigen in the organism. It is the result of recognition of antigens by certain population of lymphocytes. The process occurs at the level of organism and involves various cellular recognizing systems which are in the process of learning, since they become capable of remembering the antigen introduced for the first time, and it can also respond to its repeated injections by enhanced production of antibodies. Antibodies, it belongs to a class of serum proteins that is known as immunoglobulins. A human being, it has 5 principal classes of immunoglobulins denoted as IgG, IgM, IgA, IgD and IgE. The immunoglobulins IgG, the function, it is an function as an antibody. Their molecular weight are about 1,50,000 and the sedimentation coefficient, it is 7s. The immunoglobulins IgM are the pentamers of IgG. The structure of many IgG has been studied. In the figure 4.20, it shows schematically the structure of rabbit IgG antibodies. The molecule consists of 2 heavy chains with a molecular weight of about 450 residues designed as gamma, and 2 light chains with a molecular weight of about 220 residues designed as the alpha chain. These chains are joined by the dulcified bridges, as you see in the picture. The antibodies to the various antigens have similar structures despite the differences in the primary structure that is localized in the V region of the molecule that lies in the vicinity of N-terminal ends of the light and heavy chains. The variable regions are responsible for antibody-antigen interaction, since the antibody specificity is controlled by the specific feature of primary structure. The active center of antibody contains a variable region. These are 2 such centers in each antibody molecule. Antigens are primarily proteins and polysaccharides. Landsteiner in 1990 worked out on a method for synthesis of artificial antigens, which is based on the combination of diazotized aromatic compounds like tyrosine of the protein. Thus, any radical R can be introduced into the protein. The R phenanzaloprotein, it can be used as the antigen for stimulating the generation of respective antibodies. It has been established that the factor that determines antigen specificity, it is just the radical R, and the protein, it plays a secondary role. The antibody that is produced in the response of R-P, P stands for protein, reacts with the R-P dash. P stands for another protein. But with RP, the antigen RP produced with a respect to antibody system, the reaction is retarded when the concentration is further increased. The reaction proceeded. Small molecules do not generate antibodies in the organelles and not antigens. They interact, however, with the earlier generated antibodies, forming soluble compounds. This is called hapten action. And the small molecule compounds, are known as haptens or determinate groups. Hapten competes with the antibody places through the weak interactions that obey the mass action law. Natural antigens are multivalent, that is, they contain several determinate groups, that is haptens. The 3D structure of determinate group, it is essential for immuno process. The presence of reactive group is in antibody. It is proven by the elaborate experiments carried out by Pressman and Sternberg in 1951. A rabbit was immunized by artificial synthetic antigen that contained the p-azobenzoic acid residue or the p-azophenyl arsenic acid residue as a determinant group. The resulting agents were subjected to iodination, as a result of which their immunological activity was completely suppressed. However, when iodination was carried out in the presence of haptens, the activity was retained. Evidently, the introduction to the iodine into the active energy of the antigen abolishes its activity, and the hapten protects this region against the iodine, as you can see in the picture. uh, there is a structure corresponding between the active region of antibody. On other hand, the hapten or determinant group of the antigen. On the other hand, the structure of the complex of the antigen with the corresponding bifunctional dinitrophenyl hapten has been studied by the means of electron microscope. Figure 4.23, as you can see, it shows an electronic microphotograph of antibody DNP complex. That means dinitrophenyl hapten complex. Figure to 4.24, it is an schematic interpretation divalent antibodies interacting with the divalent DNP molecules form ternary cycles. The projection at the corners of the triangles or portion of antibodies that do not contain variable regions. When antibody is preliminary treated with the pepsin, these portions are split off, as you can see in the figure 4.23b. The equilibrium constant for the antibody antigen reactions or antibody hapten reaction are determined by carrying out experiment in vitro. Like this is the formula that is given here. AB stands for antibody and H for hapten. The dependence of K on 1 by T gives the value of H and delta S. The enthalpy changes are the orders of 10 kJ per mole, that is weak interactions of the change delta S, or as a rule positive, which may be accounted for by the liberation of hydrating water molecules during the formation of complexes. The investigations of the dependence of the equilibrium constant on the structure of the hapten or the corresponding determinants group of the antigen that has been carried out by Pauling and Pressman in 1945 to 1957. In particular, they were obtained antibodies to the ions of ortho, meta, and para azobenzene arsenate. Antibodies to each of these groups function as if they were structurally complement with the van der Waals surface of the determinant group. The dependence of the interaction to the orthoazobenzoic arsenate binds well. For example, the antibody to orthoazobenzene arsenates binds with the other ortho substituents like the radicals with the radicals OH, NO2, Cl, CH3, NH2 instead of ASO3 H negative. The less strongly metasubstituents and most weak parasubstituents. Very important is the relative arrangement of the parts of the hapten. For antibodies against para, the relative constant for binding the haptens are respectively 0, 0, 1, 0. The removal of the benzene ring from the arsenate by one CH2 group destroys the binding. The binding depends strongly on the nature of charged group. For example, the same antibody strongly binded, but they do not bind, and the iconic size is also important. These and the other data points is complementary. The structural fit between the active site of antibody and the determinant group of antigen or hapten. The study of interaction of antibody to polyalanine with alanine oligopeptides has allowed the size of active sites of the antibody to be determined by 2 into 5 into 1.1 into 0.2 nm per cube. Antibodies are globular proteins. Upon denaturation of antibodies, their ability is to bind specifically antigens and possibly to accomplish the denaturation of antibodies with the activity being recovered. The presence of hapten favored renaturation. Molecules of antibodies exhibit some flexibility, that is, the ability to undergo conformational changes. The polarized luminescence of complex of IgG with the luminescent dyes has been enabled due to the rotational relaxation to be determined. They prove to be in the order of 50 NSS. See the section 5.5. These value corresponds to the Brownian rotational motion, not on the entire protein molecule but of its small portion. That is, they are indicative of the flexibility of protein molecule. That is from my side. Thank you.